Hello everyone, my name is Raj Daman, and welcome to my first video here on YouTube. Now this is going to be the first of what I hope to be many videos on this YouTube channel, talking about different chemistry concepts and test preparation, communicating science, and academic success. Today, we're going to be talking about acid-base chemistry, a general overview of some concepts, and some typical test questions. Now let me tell you why I'm making this video. I was tutoring a student a couple of weeks ago in biochemistry. We were talking about amino acids, talking about their pKa values, and how to predict protonation states at different pHs. And what I found was we were covering concepts that my student had already encountered when she was taking general chemistry and organic chemistry, and I was showing her how we could take those simple concepts and apply them to biochemistry to solve those relevant problems. And so I figured other students could benefit from this insight. And so what can you expect from a video like this? A simple overview of concepts, where exactly they can be applied, examples of test questions that you could encounter on exams in organic, general chemistry, and biochemistry, and ultimately this video is going to help you focus your study efforts. We're not going to go through problems and specific examples in great detail. That can be covered in other videos. So let's get started. Now, acids and bases. The concepts covered in acid-base chemistry go in several places within chemistry, and it's important in several places. In general chemistry, you talk about acids and bases within the context of equilibrium, and then you're given examples about where acids and bases can be applied. For example, buffers and titrations and the generation of titration curves. In organic chemistry, you talk about the acidity of different functional groups, the pKa values of those functional groups, but more importantly, how does acidity relate to the ability of a molecule to act as a nucleophile, and how does it act as a leaving group? And this relates to, fundamentally, reaction mechanisms, how specifically reactions take place. And finally, in biochemistry, much like I was talking about in the introduction to this video, talking about amino acids and their acid-base properties. And then how do those acid-base properties relate to enzyme structure and function? So based on that summary slide, it's pretty easy to see that the knowledge of acids and bases is going to serve you well time and time again. Now before we go any further, we need to define what an acid is. And throughout this video, we're going to be using the Bronsted-Lowry definition of acids and bases. So here's Bronsted over here on the left, here's Lowry over here on the right, and these are chemists that worked independently, and through their work, they proposed a definition for acids and bases. Simply, an acid acts as a proton donor, and a base acts as a proton acceptor. Now, as I alluded to in an earlier slide, you talk about acid-base chemistry first within the context of equilibrium, because the dissociation of an acid, HA, to give H plus and A minus, this is an equilibrium process. Now, for strong acids, they really want to give up their proton. So when a strong acid is placed into water, it is said to essentially completely dissociate into a proton and its conjugate base. For strong acids, the conjugate base A minus is stable. And so if we can find ways to stabilize the conjugate base, we can then increase the strength of a given acid. So when trying to rationalize the strength of acids, what you're basically looking for are factors that can help stabilize A minus, the conjugate base. So look for different factors, such as electronegativity, resonance, or the presence of an electron withdrawing group. Now let's go back to that equilibrium process and start putting some numbers to all of this. Here's the equilibrium process. Acid dissociating into a proton in a conjugate base, and the stronger the acid, the more the equilibrium is shifted to the right-hand side. And so we can define an equilibrium constant or an acid dissociation constant known as Ka, given by this equation here. And we can see that for a strong acid, Ka is going to be a large value. Why? The numerator is going to be very large because you're going to have a high concentration of proton in conjugate base. And for strong acids, they have a large Ka value. That means they have a small pKa value. Now over here is the definition of the pKa. It's the negative log of the Ka value. And pKa values are used frequently in organic chemistry. Each functional group that you encounter has an approximate pKa value. So this brings me to one key point that I want you to take away from this video. Know your pKa values. You don't have to know pKa values for every molecule specifically, but what you want to know are rough pKa values for the different functional groups that you're going to encounter. And so you'll see pKa tables in your textbooks or in a handout, and here's an example of some rough pKa values that you could find in such a chart or table. Alcohols roughly with a pKa of 15, protonated amines roughly with a pKa of 9, and carboxylic acids roughly with a pKa of 5. If you know these values off the top of your head, you're going to make better decisions in terms of rationalizing how reactions can take place if you're writing out a mechanism, and importantly, how to rank different molecules in terms of their acidity. And it just so happens that ranking molecules turns out to be a really good test question for organic chemistry. It ties in all the knowledge about pKa's and different factors to help stabilize a conjugate base. So let me give you an example. Here are 5 different molecules, a couple of alcohols, a couple of phenols, and 1 carboxylic acid. And I could ask you simply, please rank these in the order of increasing acidity. But I could ask this in a completely different way. Could you draw the structure of each conjugate base and then rank the stability of those conjugate bases? So you'd have to take into account those factors of electronegativity, resonance, and electron withdrawing groups and make a choice as to how they stabilize each one of these conjugate bases. So that's organic chemistry. And I'm going to highlight this molecule here, acetic acid, because I'm going to bring that on over to the next slide as we transition on over to concepts applicable to general chemistry. And so here I've brought over acetic acid and its conjugate base, acetate, to help introduce buffers, something that you encounter in general chemistry. Now quickly, what is a buffer? A buffer is an aqueous solution of a weak acid and its conjugate base, or a weak base and its conjugate acid. The whole idea here is that buffers are able to resist changes in pH when a small amount of either strong acid or strong base is added. And where we talk about buffers, we also have to talk about a certain equation known as the Henderson-Hasselbalch equation. And here's the equation. It tells us that the pH of a buffer solution is going to be equal to the pKa of an acid plus the log of the ratio of A minus over HA. So the conjugate base over a weak acid. This is applied when you want to prepare buffers and in the discussion of titration curves. Here's an example of a generic titration curve here, and you can see the circled region. This is known as the buffer region. So if you want to calculate the pH in that region, the Henderson-Hasselbalch equation applies. You can use the Henderson-Hasselbalch equation to come up with different types of test questions that you can encounter on a general chemistry exam. I can give you a few examples. Number 1 is buffer preparation. I can give you the information on the right-hand side, the pKa of a particular acid, the concentrations of the acid and its conjugate base, and tell me, if you mix them together, what is going to be the pH of the particular buffer. I can also ask you to find the pKa of a particular acid and give you the ratios of HA and A minus, give you the pH of the solution in question, manipulate the equation so that you can tell me what the pKa value is. Finally, give me the ratio of A minus over HA. I can give you the pKa of a particular acid and the pH of the solution. Can you tell me what is the ratio of the protonated and the unprotonated forms of that acid? And this is an excellent test question that you'll see often in biochemistry when it comes to amino acids. So let's touch on that quickly. And here we are, amino acids. If you're taking an introductory course to biochemistry, one of the first topics you cover are amino acids and proteins. And you're learning about the specific structures of amino acids, their side chains, 1-letter codes, 3-letter codes, and of course you're talking about their acid-base properties. You learn about the pKa values of the different side chains of the alpha amino group and the carboxyl group. Here's histidine as an example. So you can highlight the carboxyl group, the alpha amino group, and the side chain. And here are the types of test questions that you can be asked. What is the overall charge at pH 6 for histidine? So what does this require? This requires knowledge of the pKa values. What is the ratio of the unprotonated protonated side chain at pH 6? The pKa of the histidine side chain is going to be given to you, and the conditions are given to you as well, pH 6. And that's a direct application of the Henderson-Hasselbalch equation that you encounter in general chemistry, directly applicable to a possible test question in biochemistry. And this is easily extended on over to peptides. What is the overall charge of a peptide at a given pH? The approach is exactly the same as with amino acids, because you want to know the pKa's of the different side chains and what are their protonation states at a given pH. And you may also encounter questions about titration of an amino acid or a peptide. So this is taking all that general chemistry knowledge about the Henderson-Hasselbalch equation and applying it within a biochemistry context. And why is this important? Consider some of the forces that you talk about that influence secondary structure of proteins, ionic interactions, sometimes known as salt bridges. If the structure of a protein changes as a function of pH, have you disrupted a particular ionic interaction? It's certainly possible. And second, the catalytic activity of an enzyme. If enzyme activity changes as a function of pH, has the protonation state of a particular residue changed? And it's quite possible. So let's recap some of the key points that we've talked about in this video. We talked about strong acids and how strong acids have stable conjugate bases when they dissociate. And what this means is that they have large Ka values, and in turn that means you get low pKa values. You find ways to stabilize the conjugate base. Factors like resonance, electron withdrawing groups, electronegativity, anything to help stabilize the conjugate base helps to increase the strength of the acid. Know, roughly, you don't have to know specifically, the pKa's of the different functional groups that you're going to encounter. This leads us into buffers and titration curves and application of the Henderson-Hasselbalch equation. And then you can take the knowledge that we gain about pKa's, different functional groups, how to use the Henderson-Hasselbalch equation. This takes us into biochemistry and amino acids. Can you predict protonation states? And how does that relate to overall protein structure and enzyme function? And I'll also give you 3 key takeaways from this video. I'll say number 1, know your pKa values for the functional groups. Know how to apply the Henderson-Hasselbalch equation, not just within general chemistry, but within biochemistry, and the examples of the test questions that I've shown you in this video. I think with these 3 points, this is going to help focus your study efforts as you prepare for exams and deal with the material in terms of acid-base chemistry. And that brings us to the end of the video. Thank you very much for watching. Please leave any comments or feedback that you may have in the comments section below. All of that is going to be very helpful as I continue to make more videos and build out this YouTube channel. You can also find me on Facebook and Twitter. The links to those pages are found below in the video description. And of course, if you need more information about me or need to reach me, my website and my email address are listed in the bottom part of this slide. Thanks again for watching, and we'll see you next time.